Hello, thanks for attending the talk. Today I'm going to talk to you about Madagascar speciation patterns, specifically the role of mountains in pelican spiders, specifically the species shown here, Eriacinus workmani. And the background to this study is a larger study that I worked on several years ago, looking across the pelican spider family, Archaeidae, looking at trait diversification patterns. And the goal of this study was to try to understand the observations that in Madagascar you have different trait diversification patterns compared to other areas of the world where these spiders occur. And so what we did is we measured some linear traits to quantify shape. These traits relate to the morphologies that directly relate to these spiders' bizarre predatory behaviors, and they have some really unusual morphologies compared to other spiders. We looked at morphospace plots. We found that Madagascar occupies a greater range of morphospace. Further, on the dated phylogeny, we showed that there were increased rates of trait diversification in Madagascar compared to the other areas, and that there was a correlation between morphology and habitat, which is suggestive that these traits are adaptive. Lastly, we looked at just patterns of the phylogeny, that you have sister species that occur on isolated mountain tops close to each other. And we found a correlation between altitude and endemicity, in these pelican spiders in Madagascar. And so the real importance of this study was that it set up a hypothesis, which is what I'm currently going to be that we're testing right now. And this hypothesis is that in Madagascar, sympatric lineages are accumulating in mountain areas. And the idea is that this is what's spurring having these sympatric communities is spurring morphological diversification in Madagascar. For this talk, I'm really going to focus on the first part, because that's where we are in this study, of just looking at how sympatric lineages are accumulating in mountains. The trait diversity part is the most interesting to me, and I'll talk about it in the very last slide just a little bit. So the focus for this study, I selected the workmani group, and that's because this is, it's a monophyletic group. It consists of 5 species. They're up in the mountains, and they have a variation in their distribution ranges. Some are widespread, some are more narrow endemics. But the important part is that the different species overlap in some parts of their ranges, and they're allopatric in others, allowing us to compare how traits are changing in allopatry versus sympatry. We needed a phylogeny to get at these questions, and so here's our phylogeny that's derived from ultra-conserved elements and mostly museum material, and it's color-coded. These are the 5 different species of the group. For this talk, I'm going to focus on just this one, workmani species, and that's because when we saw this phylogeny, we looked at it and realized we're seeing similar patterns between the species as what we're seeing within a species, in the populations in a species. So we really wanted to delve into that. However, everything we're doing with workmani we'll be doing with these other species as well. So just to show you the patterns, let's go through this phylogeny for just workmani. The first group to diverge occurs in this mountain area, sort of in the mid-eastern part of Madagascar. The next group, sister to that, is down in the mountains in southern Madagascar. Then you have this little group up in a mountain in between them. Then we have another group to diverge that's again from that purple area. The next group is from the mountains down in the far south. And finally, a third group in this purple area. These are well-supported nodes in the phylogeny. And then you have these more widespread but allopatric groups. And further, there was morphological variation in these spiders within this species, and the variation seemed to be more apparent in the sympatric populations compared to the allopatric populations. And so we just didn't know what was going on. And so our questions were, well, does this species actually consist of multiple cryptic species that are occurring in sympatry? If not, what's the level of gene flow between these sympatric lineages? And then finally, what were the historical processes that contributed to the buildup of these sympatric lineages? It was at this point that I took that UCE data set and gave it all to postdoc Nicolas Hazy. So I am the messenger for all of his results that I'm going to present for the rest of this talk. And he pulled from the UCE data, he pulled over 2,000 unlinked single nucleotide polymorphisms, and that became the data set that we used for all of the following analyses. We wanted to understand population structure, so we did a structure analysis, which resulted in these 4 different populations, color-coded yellow, purple, green, and blue. And is similar to what the distribution of these populations, similar to what we found in the phylogeny. In the far south, you have 2 sympatric populations, right down here. And then here you have 3 sympatric populations, and then these 2 up here are largely allopatric. We wanted to know whether these were cryptic species, so we used these machine learning algorithms and species delimitation. Our results, we found, recovered just 1 species. We also used multi-species coalescent models for species delimitation, and again, detected just 1 species. So we don't have a bunch of cryptic species. So then let's look at gene flow. What's going on? We measured gene flow and introgression. We found that the populations have strong genetic structure. Other than those 2, the green and blue up in the far north, the FST values are greater than 0.3, and that there's low gene flow for most of these populations, with less than half a migrant per generation. And so this is very unusual because these populations are in sympatry. And there was also asymmetric gene flow, with the yellow most southern population serving as a source, and up with the northern population serving as the sink. We tested for introgression, and we found that some populations, mainly the purple and the green, did have some introgression. And so the conclusion is that we have 1 species that maintains these distinct populations that co-occur in sympatry up in those mountain regions. So the next part of our question was looking at the historical processes. And our hypothesis is that these so these spiders live up in the cooler, higher elevation areas, and that during the Pleistocene warm interglacial periods, these populations are isolated from each other on mountaintops. Then during the cold glacial periods, these spiders can move down the mountains into the lowlands, and they can connect with these previously isolated populations, causing secondary gene flow. Then you get additional cycles, back to the warm periods, where again these populations contract up the mountaintops. And so again, you have repeated cycles of this, and you're going to get a buildup of sympatric lineages on the mountains. So that's our hypothesis, and we set out to test it. And so we use species distribution models. This is showing the current distribution. So the current populations are isolated. It shows that these current distributions are isolated in these mountain ecosystems. When we look at past distributions of the last glacial maximum, we see that during the glacial periods, the populations can move downward into the lowlands and become connected. So this supports our hypothesis about the role of mountains. We looked at demographic history, and we did find that the population sizes were reduced at the end of the last glacial maximum. And this is again congruent with our hypothesis and with our species distribution models as the populations contract up in the mountaintops. We did another analysis also looking at demographic history, and we found that populations declined and diverged congruent with interglacial warm periods, and also that there was secondary contact later and asymmetrical gene flow from the south to the north, in line with what we were finding in our previous analyses. And so this supports this idea that the mountains, it is congruent with our hypothesis about the mountains building up species. So our conclusions are that Workmani is 1 species with genetically differentiated populations that seem to be maintaining their distinctness even though they're in sympatry. and that the glacial cycles due to habitat contraction and expansion are causing the buildup of sympatric species in the mountains. We also conclude that speciation is a process, and we would like to suggest that we're somewhere in the gray zone of this process with this species. So either these populations are coming together, or maybe they're ultimately going to diverge and become different species. And so lastly, I want to talk about what's, I think, the most interesting part of this study, which is what we're just beginning to get into. So this is all preliminary. But we're going to go to Ranamafana Forest in Madagascar, and this is where we have those three sympatric populations that co-occur in sympatry, and just look at the morphology. And so the first obvious difference is that these three populations differ in body size, so in scale. You have a large population, a medium, and a small. And it doesn't seem to be continuous. It seems to be discrete. And so when you pull from the phylogeny, right, we're sampling one of the big, the medium, and the small to assemble this community in this one area. And of course, the big question is, did these size differences evolve in allopatry or after secondary contact? And that's what we're working on. We need to do fine scale, detailed morphological analysis, not just body size, but other traits, and the function of the jaws and the chelicerae, which will all be done in the next few years. Lastly, I want to say that there are also additional Workmani group species that occur in this area that have two populations that seem to be genetically distinct, the same pattern, except they're not differing in scale like what we see in Workmani, but instead they're differing in the shape of the carapace, with one species getting sort of a longer neck and jaws compared to the other. And so there seem to be different ways for these spiders to coexist in sympatry, and our hypothesis is this relates to their sort of niche and the prey that they can target. But again, we're still testing this. Further, things can get quickly out of hand because there are additional pelican spiders in Madagascar that coexist with these. And however, we think that ultimately at the end of this study, in several years, we hope to elucidate how mountains play a role in accumulating sympatric species, and more importantly, how that relates to diversification of the group. And with that, I would be happy to take any questions. Yes.
Thanks so much for the talk. Did you test some isolation patterns in those populations, and maybe you think about the flickering connectivity about them?
You're asking if I tested for how isolated?
Yeah.
I think that, so again, the postdoc did these population genetic analyses, but my understanding is that div migrate does measure connectivity and gene flow and how isolated they are, and we found that the southernmost population, the yellow, color-coded yellow one, is the most isolated, and that one is sort of, it is sending migrants up north, but there's nothing really coming back, but it is the most isolated. Does that answer your question?
But didn't try something like, for example, isolation by environment or distance or
Oh, we did. I didn't show by distance. We do have isolation by distance, and I took that slide out because of time. But we did test for isolate, and we do show that there is a pattern of isolation by distance. Yep. Any other questions? Okay, I think the next person can come up and get their talk ready.
Hello, everyone. I'm Ethan. I'm from the University of New Mexico, soon to be Texas Tech, and I'm going to present on some work that we've been doing looking at comparative phylogeography in the Solomon Islands in collaboration with our collaborators at the University of Kansas. So when I talk about island bird radiations, a lot of people think about these adaptive radiations, these famous Galapagos finches and Hawaiian honeycreepers, but in reality, a lot of Pacific Island avifauna looks more like this. They are comprised of non-adaptive or geographic radiations that show patterns where they're spread across these different archipelagos, with each archipelago having its own clade of, say, whistlers here, and those whistlers differentiate among those islands. And this is a pattern that's been studied a lot in the past that contributed a lot to allopatric speciation theory, particularly in the Solomons. They're this archipelago to the east of New Guinea that was featured very prominently in a lot of Ernst Mayr's work, particularly during the modern synthesis times, and has been studied a lot past then as well, such as in this great Birds of Northern Melanesia book that compiled a lot of that geographic data, and we actually pulled some of that for this study today. It's also been a target for a lot of recent resurveys by natural history collections, which I want to mention up front, provided all the tissues that were used in this study and are super essential for this kind of work. Part of the reason everyone's been so interested in it is that it has a lot of endemism, actually the highest of any endemic bird area, and that's due to its geography at two scales. First, its relative distance from New Guinea makes it able to get birds moving from that continental area, but not so much that they genetically swamp them. And it's also got a lot of geographic structure within it. You can see here these 4 island groups that we're going to be talking about throughout this presentation. The Bukita group is this long one that was largely connected during glacial cycles. That's what that dark area is. It runs parallel and is relatively close to Malaita and the New Georgia group, which allows it to likely more readily exchange genes with them. And finally, there's this isolated island of Makira on the far end of the archipelago, which is both slightly further from the rest of the islands and is in an unfavorable orientation, right? It's not running parallel, unfavorable orientation to receiving migrants. And that leads to the first of 2 questions that we're going to ask about how gene flow and dispersal play a role in archipelago evolution. This first one's actually going to start with phylogenetic inference as kind of a framework for thinking about how these taxa evolve. A lot of birds start on the west side of the archipelago, and what we showed using simulations with SLIM, with input from Pleistodis, my lab-made software, is that you have, when we do these geographic simulations, you pretty much exclusively have Makira as the last island colonized, which should produce a pretty predictable phylogeographic pattern. I've got that whole thing simplified here. Remember, purple is Makira, where we expect Makira to be well nested within the phylogeny, and the New Georgia group primarily to be sister, regardless of the precise colonization direction. But we oftentimes see, both in phylogenetic studies that have been published, in addition to just the general distinctness of taxa, that Makira kind of stands out. And this topology, obviously, is not consistent with our simulations of geographic colonization, following a stepping stone model, but it is consistent, potentially, with that relative lack of gene flow that Makira receives from the Bukita group in particular. The other question that we're going to look at relates to this paradox of the great speciator. Pretty much it's the idea that if you have too much or too little dispersal, you're not going to get strong geographic variation, right? This is because of that dual role that dispersal ability has in both gene flow and colonization between islands. If you've got too much dispersal, you're not going to get any geographic structure. If you have too little dispersal, you don't have any geographic structure, and you're not even going to see those in these studies, of course, because it's all about the comparative side. And then if you have just the right amount of dispersal where you can get to new islands, but you're not getting panmictic once you're there, you can have this incredible geographic variation that's been the inspiration of so much work in this area. A lot of our past work in this system, I'm briefly going to touch on, addressed more that kind of topology side. First with a case study in these black and white monarchs, which were a really well-sampled group in the archipelago, where we found that Makira sister pattern, in addition to evidence of gene flow supporting our hypothesis of Makira's relative isolation. We also ran a bunch of simulations to support our conjecture as well, and found that we are able to produce the topologies that we see with the colonization history that I discussed, with Makira getting colonized last. And not just did we didn't only recover this topology, I'll note this was the minority in the simulation that it came up in, but it took very little gene flow, like way less than 1 migrant between island groups in the whole archipelago, to erase the initial signal of colonization, which I found very concerning in general. But what we really are going to talk about today is this comparative study that we're doing, where we're testing the generality of these findings and then addressing some new questions that we can't look at with just one taxon. So we added 10 more. There are 4 existing data sets. As of this morning-ish, it is now 2 published and 2 in prep. In addition to 6 new ones, all this is in collaboration with folks at the University of Kansas. And they have even more that is coming with that we're going to incorporate into this larger data set. So we did RAD-seq on all these, I forgot to mention. And we calculated a series of population genetic and phylogenetic summaries. The main one that we're going to focus on today is the divergence between islands, FST. We also looked at nucleotide diversity. That's not going to come into play today. That's in our full models, and also that phylogenetic component. And then we added to this a few ecological metrics. The main ones that we're going to focus on, in concordance with that paradox of the great speciator, is dispersal ability. We looked at 2 different metrics that we say more or less represent realized dispersal, the number of islands occupied, and ability to disperse, hand-wing index, pretty much how good a bird is at generating lift. What we are asking with these, of course, is our inference question. Is the Mercuric sister pattern more common than we would expect? Pretty much confirming that what we saw in those monarchs wasn't an outlier. And then ask some questions about differentiation, both if great speciators show significantly higher genetic divergence, and what broadly predicts genetic differentiation in all these taxa. Starting with our topology question, we found that the Mercuric sister pattern was common. 4 of 7 well-resolved topologies, 3 of 5 where we are very confident in the colonization direction that occurred, which is statistically significant with a binomial test. But there were no strong predictors of what was Mercuric sister versus otherwise well-resolved topologies. Now getting to what I find most exciting about this, these predictors of divergence patterns. First, we found that great speciators were not statistically significantly different from all of our other taxa in terms of either on the left, our metric of genetic divergence, or on the right, our metric of dispersal ability. This is in part because one of the great speciators is relatively weakly diverged, and there's several that don't show super strong geographic variation in plumage that led to them being upgraded to the lauded great speciator status, but they do show strong genetic divergence despite that. I'll note that super high FST is the one that just had a paper come out, the greatest speciator. Anyways, as for our modeling of what metrics best predict this inter-island group divergence, again, this is, say, divergence between Bukita and New Georgia Makira, et cetera. We took the average of all of those different combinations to get this. We found that the number of islands occupied was consistently the best predictor of it, in pretty much any way that you can pick out your favorite predictor, which, again, we got from that Meyer and Diamond book. It's a coalition or combination of a bunch of surveys that occurred over decades of these islands, and we think it represents kind of realized dispersal ability, the ability for them to cross water gaps, reach smaller islands. And then hand-wing index was also a very strong predictor. That's that dispersal ability. But of course, they are very highly correlated. This is all pretty cool. Even just this result that they are correlated is a good demonstration of hand-wing index for measuring dispersal ability in these taxa, and then that impact down the line on genetic differentiation. All of this, in addition to getting these cool metrics and answering our big hypotheses, led to kind of a more qualitative thing of how we think these taxa have evolved in the Solomon Islands, which we hypothesize starts with colonization and a relative genetic bottleneck, but notably with strong clustering and a rapid time, because in order to get to the Solomon Islands, you have to have a pretty decent dispersal capability, right? It's pretty distant from adjacent archipelagos. But once you're there, the islands are relatively close, which we think allows birds to quickly colonize between them. Then the populations equilibrate, get back to reasonably large effective population sizes, potentially with weak clustering corresponding to these relative levels of gene flow, which over longer periods of time, particularly if dispersal ability reduces over time, which we have seen in many cases in birds that it does through various lines of evidence, you could get a topology strongly supported that is not based on biogeographic history, but rather is just based on the relative gene flow over time in the archipelago. To wrap it all up, we found that our Makira sister hypothesis held for a good amount of taxa, and it led us to a hypothesis for how we get there. We found that great speciators don't stick out relative to other taxa, at least not statistically significantly, other than in the number of subspecies. And metrics of dispersal were the best predictors of genetic divergence in our archipelago. So I would like to acknowledge the folks that made this possible, of course, the museums that provided all the tissues, and also particularly the Museum of Southwestern Biology, where I measured a lot of the hand-wing indices. The folks in the Solomon Islands that allowed new collections to occur, and both the Moyle Lab at KU and Anderson Lab at UNM that are collaborators on this, and the UNM biology group generally for providing feedback. And of course, our computational resources and funding. And with that, I'll take questions.
Thanks, that's really interesting. Apologies if I missed this, but how are you defining your great speciated?
This was based on I'll go to the mic. This was based on categorizations that ornithologists, namely Meyer and Diamond, did in that book. So that was taken from that compendium book. That itself was based on the number of described subspecies, which we take as a proxy for the ability of ornithologists to parse out the geographic variation, which clearly is not sufficient for describing any genetic process. Yeah, and a lot of that is, of course, plumage variation, because ornithologists love their plumage. Anyone else? All right, thank you very much.
Good morning. It actually feels like midnight for me because I've come from a very, very long way away. And I will admit that I often don't come this far to go to a conference. So I want to spend, rather than dive straight into the science, I want to just tell you a little bit about the place where I live and work. Because I'm assuming that many of you might not have actually been to Australia before. But you know, even if you're not that familiar with the land down under, I think you probably know more than you think you do. So, nothing is going to stop working. There we go. So, if you're interested in human history, you might have an appreciation for the Indigenous Aboriginal people that have been the traditional custodians of Australia for more than 50,000 years. So, in Australia, we have the oldest archaeological evidence of modern humans outside of Africa. And that map that you can see there is the different language groups of Aboriginal people, and you can see that it is staggeringly diverse. If you're more into sort of modern language and you find my accents lovable, then you might know Australia is a land of g'day and just all sorts of other very bogan words that I've put up there. And actually, Australia is probably the only part of the world where you can ask someone to take a squiz in your pigeonhole and not get arrested, which is something that Craig Moritz famously did when he was at Berkeley, actually. You might love Australia for our arts and culture. Or you might love watching paint dry. And if you have 5 days to lie on the couch, cricket might be your favourite sport. This person who wrote that little sign down the bottom was so bored they don't even know how to spell the word cricket. But even if none of those things interest you, you're probably familiar with the enormous and unique diversity of Australia's endemic plants and animals, which is in part due to the famous voyage north that the continent made some 40 million years ago when it separated from the rest of Gondwana. So the plants and animals that were already on this migrating continent had something like 20 million years with which to diversify in relative isolation. And our remarkable marsupial radiation is a famous example of that. But lots of other things have radiated spectacularly and rapidly in Australia. We have a crazy diversity of lizards, especially in the arid zone. In fact, we have maybe the highest diversity of lizards anywhere on the planet here. And I am a herpetologist. This is a group that I know most about. I am a herpetologist at heart. But I'm also a geologist at heart. And I love Australia because 70% of it is a desert, and I love deserts. And the Australian desert is extraordinary because it is enormous, it is biologically diverse, and it has been there for hardly any time at all. So today, I want to talk to you about what we can learn about the evolution of Australia's arid zone by looking at the evolutionary history of the organisms that live there through the patterns in their DNA. And I sort of, this sort of theme that I've worked with for a long time is a little bit different to what people traditionally do. So a lot of you people in the room, you're phylogeographers, we use information from geology and geomorphology to kind of help us understand the patterns that we see in different taxa. And sort of the things that underpin those patterns. But most of Australia's arid zone is actually buried under 30 metres of sand. And the deserts are super remote, and so geologists don't really know very much about them. So I'm interested in how genetics can inform earth sciences about how and when those deserts formed. And if you're interested in this, I encourage you, it's very long, but I encourage you to read this paper that I wrote while we were in lockdown a few years ago, which kind of puts the geological and climate literature into easy to understand language for biologists, and sort of sets up a series of hypotheses that could be tested about the sort of the ways in which geological processes might drive evolutionary processes in the deserts. Oh God. Ha. Okay, so Australia is really, really old. It's actually got it's got the oldest landscapes in the world in Western Australia, actually. And it's been tectonically stable for a long, long time. So those green stars there, I've highlighted where we have our mountains or uplands. I use that term very loosely because most of you, we say mountains and you think of things that are 5,000 metres or taller. In Australia, all these areas are less than 2,000 metres and some much, much less than that. And they've been tectonically stable for like 60 to 80 million years. So nothing has happened there for a long time. In complete contrast, the deserts are super, super young, maybe at most 1 million years old, and most of them are thought to be much, much younger than that, formed in the late Pleistocene when the glacial cycles were deeper and more arid. And actually, the deserts during glacial maxima, the arid zone was actually much, much bigger. So we have evidence of sand dunes right on the edges of the continent. So, at the LGM 20,000 years ago, it was 9 degrees colder, it was very, very windy, there was areas of mobile sand dunes. So as you can imagine, a change like this across such a huge part of the continent would have had profound consequences for anything that lived there. So when you think of a desert, when I think of a desert, a lot of people think of something like this. So parts of the Sahara look like these gigantic mobile sand seas. That is not at all what Australia's deserts look like. For a start, our deserts are quite vegetated and we have very few active dunes. I'll give you a little bit of a slideshow because you probably don't get to see this stuff very often. Most of our deserts have dunes that are, long, tens of kilometres long. They're 5 to 25 metres high, spaced about 200 to 600 metres apart. So this is a picture from the Simpson Desert. This is the Great Sandy Desert in the west and the Strzelecki Desert. We have this part of Eastern Australia called the Channel Country. It's a big ancient floodplain and there are gigantic sort of ephemeral river systems that most of the time are dry and look like that. But every now and again we get really good rain up north. The Channel Country, these big river systems flow. They flow through the deserts, they flow through the dunes. And when that happens, we all go, wow, it's so spectacular, it's so rare. But that's actually much more like the environments that most of the arid zone organisms have evolved in for their deeper history. So we know from the most well-sampled dunes, if you can see the red dots there on the left of the map on the left, the most well-sampled dunes, the dates for those are concentrated in the last glacial cycle, so at the LGM. But we really don't have very many dates from anywhere else in Australia's deserts, and it's obviously quite big. So I want to know, can we get some idea about the relative ages of these different deserts based on the timing of population expansions of desert taxa? And I read this really cool paper by Ariana Kuhn, and she was interested in synchronous expansion of the herpetofauna of Madagascar. And I thought, well, this would be a really cool way to look at, can we look at synchronous expansion of arid zone taxa using the same method? And it just so happened that last year we had this new program in our research school of biology where we could get an Indian undergraduate student to come over from India and work on a project with us. And so that's how I met Advait, who came from the Indian Institute of Science. And he was a third-year undergrad. He arrived and I was like, okay, Advait, what are you interested in? And he's like, I'm interested in the behaviour of social insects. I'm like, that's great. How about a project on mitochondrial genetics looking at demographic reconstructions of arid zone vertebrates? And he was like, okay, I'll do that. So he was totally awesome. So when I say like we in this talk, it's totally he. And we also had a lot of input from Ian Brennan, who was a postdoc in our lab at the time too. So I don't know about you, but I and a lot of my colleagues, we have all these unpublished mitochondrial data sets which we don't know what to do with, just sitting there. So we talked to a lot of people and looked in our bottom drawers and we found a whole bunch, and also went to GenBank. There's just so much mitochondrial data that's available now. So we took all that and then we wanted to use Ariana's method and to see what we could come up with. So I'm not going to go into any detail really. You can read her paper, it's really great. But basically you make a bunch of trees using BEAST. We identified the panmictic populations using BGMYC, and then we want to determine which of those populations are expanding. When we did that using 3 different things, it was ABC, neutrality statistics, and Bayesian skyline plots. And then once you have your expanding populations, you throw them all together and use a program called PipeMaster to look at synchronous expansion events across multiple populations. So as you can see, we had a lot of populations. So, and that's not even all the data. There's actually ones that Advait hasn't included yet. So these are the results from the single population ABC analyses. And that plot there is the demographic probabilities that were assigned, that each of the populations was assigned to. So either expanding, constant, bottleneck, or a bunch of indeterminate. And you can see that there's like a lot of expanding populations, so 199. So we've got a lot of data to work with. This is where I apologise and say I greatly overestimated my ability to be able to go any further on this project, because actually Advait had to go back to India. We're still working on it, but we actually haven't thrown them all in together. So I just want to show you what we want to do once we actually do the synchronous part of this project. So basically, not only do we want to look and see whether or not, if you throw everything in together, can we see synchronous expansion? But if we break it down into different areas of the desert, the eastern deserts versus the western deserts, for example, they have very different histories. Can we tell whether some are older than the other? We can also break it down phylogenetically. We can look at whether or not there's different responses, for example, between reptiles and mammals. So this is one way you can look at the LGM and its impact on demographic history, obviously just using one gene. There are lots of other different methods and different approaches using different kinds of data. My postdoc at the moment, I've been collecting SNP data for a whole bunch of arid zone reptiles. So here you can see effective population size estimates using EPOS. This is like a method that uses site frequency spectrum. And you can see that it actually, this method requires that you have very little population structure. You need at least 30 individuals, you need at least 5,000 SNPs. But you can see the resolution at the LGM, like around that 20,000 years ago, is quite good. It's picking up expansion there. It's great. But the problem with these SFS type approaches, there's EPOS, there's stairways too, they're a bit limiting because you need lots of individuals. And this sort of data that I have hasn't been collected sort of for traditional population genetics. I don't have that kind of, often I don't have the sample sizes that are necessary. There are other SNP-based approaches. You can use like something like GORN, which is based on linkage disequilibrium, but you can't use that if you've only got anonymous SNPs and if you don't have reference genomes, and I don't have that. So there are also genome-based approaches for reconstructing historical demography. You've probably heard of PSMC, which is a coalescent-based approach that just uses one individual. So that's great because you can get around the sample size problem, but it's not very good at reconstructing things in more recent times, like at the LGM. But MSMC is like the multiple sample version of PSMC, and this is much better at reconstructing recent things. And this is just a figure from my friend Emily Roycroft. So she's been looking at using MSMC. This is sister pairs of geckos. She said that she used 5 individuals, so that's 10 haplotypes each, and she's getting really good resolution all the way down to 1,000 years ago. So this is going to be perfect for looking at the LGM. So whole genomes is clearly the way to go, but there aren't very many yet, and especially for Australian taxa. So this, well, you can't even read those, so there's no point trying to tell you about that. But there is an initiative that I've been part of, AUSARG, the Australian Amphibian and Reptile Genomics Initiative, and as part of that, we're generating 10 reference genomes across across reptiles. There's some more for amphibians, but none of them are closely related to anything that I have SNP data for. So that's what I'm going to be doing, because I just got some funding to do reference genomes myself with genome resequencing for lots of individuals for each one of those species. So yes, I hope I've convinced you the Australian deserts are really interesting, and that their recent emergence across most of Australia was one of the biggest and quickest changes that the landscape has ever seen. And I think mitochondrial DNA is still, I think it's just super, super powerful just in terms of how much of it is out there. So it'll be interesting to see how it compares to the whole genome stuff when we can do that. Anyway, thank you.
All right, good morning, everyone. My name is Elyse Parker. I'm a postdoc in Mark Westneat's lab at the University of Chicago. Following up on that great talk about Australian deserts, I'm actually going to talk about a cold desert, specifically the waters around that cold desert, and I'm going to be discussing the comparative phylogeography of Antarctic notothenioid fishes. So I don't need to tell anyone in this room that anthropogenic climate change is rapidly altering marine environments, leading to increased seawater temperatures, changes in ocean currents, as well as rising dissolved oxygen levels. As a result of these and many other threats, it's become more crucial than ever for us to strive toward a deeper understanding of the oceanographic and climatic factors that promote the generation, distribution, and maintenance of marine biodiversity. The geographic and environmental features that influence diversification are frequently identified through comparative phylogeographic studies, in which patterns of genetic variation are compared across multiple co-distributed taxa, and when these co-distributed taxa exhibit shared patterns of variation, it becomes possible for us to identify features of the landscape that may play an important role in shaping these shared patterns. But of course, comparative studies can also reveal contrasting patterns of population structure. For instance, while the Indo-Pacific barrier may have played an important role in the divergence of these 2 butterfly fish species, there are multiple species of unicorn fishes that actually exhibit high population connectivity across the same barrier. And this raises the question of whether contrasting phylogeographic patterns reflect intrinsic differences in organismal traits. So, in order to better understand how patterns of diversity are shaped by climatic and geographic factors, we should figure out how to incorporate organismal trait data into our traditional comparative phylogeographic frameworks. The Antarctic notothenioid fishes provide a really excellent empirical system for doing this. So they're this really iconic example of adaptive radiation. They include more than 80 species in Antarctica that occupy a wide range of ecological niches, and they dominate the species diversity, abundance, and biomass in the region. Interestingly, many of these species exhibit overlapping, widespread circumpolar distributions, and a major question in Antarctic science concerns how genetic variation within each species is spatially distributed around the continent. The prevailing view is that notothenioids generally have high population connectivity as a result of both the unique physical features of the Southern Ocean, as well as the unique life history characteristics of notothenioids. So specifically, the Southern Ocean, as you can see in this map, is characterized by multiple circumpolar currents, most notably the Antarctic circumpolar current, indicated by the red arrows, and these currents have the potential to carry pelagic organisms across really long distances. Additionally, the pelagic larval stage in notothenioids is particularly long compared to other marine fish species, providing ample opportunity for dispersal of these pelagic larvae across the continent. In accordance with our expectations, multiple studies based on both Sanger sequence loci and genome-wide SNP data reveal high population connectivity and no evidence of genetic differentiation across the continent for multiple node attenuates, including the commercially important Antarctic toothfish and the Antarctic silverfish, which is really abundant in pelagic habitats. However, there are a handful of studies that have recognized strong geographic structure in some node attenuates. For example, the emerald rock cod, as you can see, is pretty structured between the Ross Sea and the Antarctic Peninsula regions. And so this raises the question of whether there are certain intrinsic biological characteristics that explain contrasting patterns of gene flow and structure. For instance, the Antarctic toothfish, this was that non-structured species, exploits the pelagic zone, whereas the emerald rock cod is found in benthic habitats, and these differences in water column usage could be driving the different patterns of population connectivity. Unfortunately, though, most of the phylogeographic studies we have for node attenuates at a continental scale have really only focused on one species at a time, and this lack of a comparative framework prevents us from understanding how species with different ecologies or life histories are responding in concerted or idiosyncratic ways to the shared physical features of the Southern Ocean. So that's what I wanted to do here, is compare patterns of population connectivity across multiple widespread circumantarctic node attenuates and ask whether there are particular life history or ecological traits that may predict the observed patterns. To address these questions, I collected genome-wide SNP data using dd-RADseq for 8 node attenuates that exhibit these circumantarctic distributions, notably 2 species belonging to the icefish clade, which you might know as those fish that lack hemoglobin and red blood cells. 2 species of the notoperch clade, Trematomus, which are really abundant in Antarctic shelf habitats, the Antarctic toothfish and the Antarctic silverfish that I mentioned earlier, the barbelled plunderfish, and the gray rock cod, Lepidonotiths squamifrons. These 8 species were collected because they really represent nearly the full range of variation among node attenuates and some of these ecological and life history traits that may influence dispersal capacity. And that allows us to test a wide array of different biological traits and their influence on phylogeographic patterns. One quick caveat is that obvious challenges with sampling in this region mean that I don't actually have circumantarctic distributions. So we're really looking at patterns of connectivity and structure between 2 regions, the Antarctic Peninsula and South Scotia Arc in purple, and the Ross Sea region in orange. To address our first objective, I employed a principal components analysis of the genome-wide SNP data. I hypothesized that if populations from the Peninsula and Ross Sea region are genetically distinct, we should see non-overlapping clusters of individuals corresponding to those regions, whereas significant overlap of clusters will be interpreted as evidence of a lack of genetic structure. For the icefish species and the 2 pelagic species, the Antarctic toothfish and the Antarctic silverfish, we do see near complete overlap between the 2 regions in the genotype space, reflecting a lack of genetic differentiation between them. But for the barbelled plunderfish, the 2 Trematomus species, and Lepidonotiths squamifrons, we see pretty clear separation of populations corresponding to the 2 regions along the first PC axis of the genotype data. And this lets us know that there is some geographic structuring between these regions. As a complement to the PCA, I used a genotypic clustering analysis, SNMF, which is analogous to structure, and that we can first estimate the number of genetic clusters in our data and then probabilistically assign individuals to these clusters. In general, our results are consistent with the results of the PCA. So for the 2 icefish species, the Antarctic toothfish and the Antarctic silverfish, an assumption of 2 genotypic clusters does not correspond to clear genetic distinctions between these 2 regions. So instead, both genotypic clusters that we identify harbor individuals from both geographic regions. On the other hand, we do find evidence of some significant geographic structuring within the barbelled plunderfish, the 2 Trematomus species, and Lepidonotiths squamifrons, with the majority of individuals from the peninsula assigned to 1 genotypic cluster, and the majority of individuals from the Ross Sea assigned to a 2nd cluster. Collectively, the results of the phylogenetic or the phylogeographic analyses reveal evidence of contrasting patterns of phylogeographic structure among node attenuates, ranging from virtual panmixia to strong geographic structure. And this, I do want to quickly just make a caveat. So as you can see, the sampling here means that we can't rule out that there's some isolation by distance happening between these populations. It's not something we can test, but regardless, we're still seeing pretty strong differences between our node attenuate species in terms of the spatial distribution of genetic variation. This finding led us to question whether there are intrinsic differences among these species that might underlie the contrasting patterns that we're observing. To address this, I applied a random forest classification, which is a machine learning approach in which you build multiple decision trees that classify a set of observations to a set of response categories given a set of predictor variables. In this case, our response categories represent whether species are structured or not structured based on the results of the population genetic analyses. I then conducted a review of museum records and the primary literature to collect data on a suite of ecological and reproductive traits, and these represent our predictor variables in the classifier. So among these are things like maximum fecundity, and this is absolute fecundity, average egg size, maximum body size, mean percentage buoyancy basically reflects different usage of the water column, and we also looked at multiple measures of depth occupancy. Overall, the predictive accuracy of the classifier was fair at about 63%. Our cross-validation approach correctly predicted which species were non-structured with 100% accuracy, and only 2 species were incorrectly predicted as structured rather than not structured. We identified the most important variables for predicting population structure by first sequentially removing each variable from the classifier and then calculating the mean decrease in accuracy of the classifier with that variable removed. And that revealed 3 traits that were pretty important in predicting population structure. These were maximum body size, average egg size, and maximum fecundity. So maximum body size kind of makes sense in light of the long history of research on marine organisms that show that larger body sizes tend to confer higher dispersal capacity. With average egg size and maximum fecundity, these kind of go hand in hand. We expect that things that have lower average or higher average egg size and lower fecundity, like the structured species, are generally going to have more investment in the offspring and thus potentially a higher probability of survival of those offspring along their long journey from one locality to the next. So these results are pretty consistent with what we might expect with regard to dispersal capacity in marine fishes. There are a couple of notable exceptions that you might see. So the Antarctic silverfish have a really small maximum body size, a pretty small average egg size, and their maximum fecundity is like roughly on par with that for the non-structured species. But I want to mention that pretty much all notothenioids or most notothenioids are nest garters or exhibit some form of parental care, whereas the Antarctic silverfish is a broadcast spawner with pelagic eggs, so it has this very unique life history relative to other notothenioids, and their pelagic eggs are likely getting advected into the current and carried around the continent. Additionally, the Antarctic toothfish exhibits relatively high fecundity, as you can see here, but I want to mention again that I measured absolute fecundity because this was the only measure that was available for all of the species I looked at. If you look at relative fecundity, which takes into account the maximum mass of the species, the relative fecundity of toothfishes is pretty small. So this species looks like an outlier, but it's not really. So despite these exceptions, we do identify a handful of traits that seem to be useful in predicting whether notothenioids exhibit population structure or not. So to conclude, I did identify evidence of contrasting patterns of population structure between the Antarctic Peninsula and South Scotia Arc and the Ross Sea, ranging from virtual panmixia to pretty strong geographic structuring of populations. The results of our random forest classifier indicate that contrasting patterns of phylogeographic structure can be predicted by body size and differences in reproductive output among notothenioids. Finally, I want to point out that this trait-based phylogeographic approach is useful not only for revealing which traits are most important in explaining observed phylogeographic patterns, but also for predicting patterns of population structure and species for which we might be lacking population genetic data. And these investigations are becoming increasingly important in light of the growing anthropogenic threats to biodiversity. So in this study, we've identified a suite of traits that can be used to identify or predict patterns of genetic diversity in a group of marine fishes that are really hard to sample and live in a really imperiled area. I'd like to acknowledge the following people, and with that, I'll take any questions. Oh, Prasanta, yes.
Sometimes it seems like in the Ross Sea there are these like huge populations of nesting.
Yes.
Anyways, like do you see that in other species, or can you predict that they might be doing that based on someone's biology?
So it's hard to, on the phylogeography, predict whether or not some of them are going to be exhibiting that nest guarding behavior, but we can kind of predict it based on that ratio of like egg size and fecundity. It does seem like most nodothenioids are exhibiting that behavior with the exception of a few broadcast spawners, but yeah, it's something that needs to be observed for more species, for sure. Thank you. All right, thank you.